Once it was proved that DNA was the primary genetic material in the living system, it was natural to conclude that DNA replicates as a molecule, qualifies as genetic material only if it replicates. Scientists Watson and Crick, who in 1953 had proved that DNA had a double helical structure, had also suggested a probable scheme for the replication of DNA. According to their proposed scheme, the two strands separated and acted as a template for the synthesis of two new complementary strands. After the replication process was completed, each DNA molecule would end up with one parental strand and one newly synthesized strand. That is why this DNA replication scheme was referred to as semi-conservative. In 1958, to find conclusive evidence for this scheme of semi-conservative DNA replication, experiments were conducted by scientists Matthew Meselson and Franklin Stahl on bacteria Escherichia coli or E. coli, a bacteria that divides every 20 minutes. Meselson and Stahl grew E. coli in a medium that had NH4Cl15 isotope as the only nitrogen source. N15 isotope is a heavy isotope of nitrogen, but it is not radioactive. Many generations of E. coli were bred in the same medium, which was not too hard to achieve since a new E. coli generation arrived every 20 minutes. As a result, newly synthesized DNA too had the N15 isotope as a constituent of purines and pyrimidines. This new heavy DNA molecule was distinguished from normal DNA by virtue of its high density while being extracted via centrifugation in a cesium chloride or CsCl density gradient. Later, E. coli cells were transferred into a medium with normal NH4Cl14 isotope. After the cells multiplied, their DNA was extracted and separated independently in CsCl gradients to record the density of DNA. It turned out that DNA extracted from the first progeny of E. coli after the transfer from the N15 isotope to the N14 isotope medium had an intermediate density. It was referred to as hybrid DNA. The next generation of E. coli, which was born after 40 minutes, had equal amounts of hybrid DNA and light DNA. Thus, the findings of the Meselson and Stahl experiment established the validity of Watson and Crick's theory of the semi-conservative DNA replication scheme. Similar experiments were further conducted in higher organisms such as plants and human cells to prove the same. One such experiment was conducted by Dr. J. Herbert Taylor and his colleagues in 1958. In root tip cells of Vicia faba or faba beans, using radioactive thymidine to determine the distribution of newly synthesized DNA in chromosomes. Here again, the experiment proved that DNA replication even in chromosomes was semi-conservative. Therefore, with multiple experiments conducted by different scientists, it was concluded that DNA replicates semi-conservatively.